Welcome to Geology 108, Precambrian life. Life most likely started during the late Hadean or early Archean Eons. It took at least 3 billion years, most of Earth's entire history, for the first unicellular organisms to evolve into animals. In this important period of life history called the Precambrian, prokaryotes learned to derive energy from the sun and gradually produced Earth's tertiary atmosphere by exhaling oxygen as a byproduct of their metabolism. Eukaryotes developed respiration and invented sexual reproduction. Soon, eukaryotes learned biological cooperation and built the first multicellular organisms. As Earth's climate stabilized, this was rapidly followed by a period of intense biological diversification called the Cambrian radiation. In this lecture, we will narrate how life came to be and evolved during the Precambrian period. Although the origin of life on Earth is unknown, hypotheses include a chemical origin in the early atmosphere and ocean, deep sea hydrothermal vents, volcanic zones, and delivery to Earth by comets and other objects. The early atmosphere and ocean were very different compared to today's. The oxygen-free atmosphere produced a reducing environment with abundant methane, carbon dioxide, sulfur, and nitrogen compounds. This is what the atmosphere is like on other bodies of the solar system, like Venus, for example. Life could have sparked from the chemical reactions at the interface between the atmosphere and the ocean, a setup that was reproduced in the Miller-Urey tabletop experiment. After ignited sparks within a vessel filled with water, methane, ammonia, and hydrogen, amino acids started to form. Amino acids are the fundamental building blocks of proteins and therefore life. The discovery of an isolated ecosystem around hydrothermal vents on deep sea mid-ocean ridges provided another explanation for the origin of life. The hydrothermal vents have a unique ecosystem of critters with chemosynthesis as a foundation of the food chain instead of photosynthesis. The ecosystem is deriving its energy from hot chemical-rich waters pouring out of underground columns. This suggests that life could have started on the deep ocean floor. Another possibility is that life or its building blocks came to Earth from space, carried aboard comets or other objects. This is the panspermia hypothesis. Amino acids, for example, have been found within comets and meteorites. This intriguing possibility also implies a high likelihood of life existing elsewhere in the cosmos. Despite much scientific progress, the emergence of life still eludes us because there's no modern equivalent, and scientific experimentation cannot reproduce the enormous span of time and space involved in nature. Regardless of the possible origin of life, all live organisms today are intricate collections of chemical reactions organized within a cell enclosed by the protective layer of a membrane. Cells are made of organic molecules, that is, carbon-based molecules that include the carbon-hydrogen bond, and biopolymers, including proteins and nucleic acids. The membrane of a cell is a thin layer of lipids with proteins embedded in it that separates the materials inside from the outside environment and facilitates the controlled exchange of materials and energy between the cell and its environment. Many bacteria are unicellular, consisting of just one cell, but most organisms are multicellular and consist of hundreds to trillions of cells. In multi-celled organisms, some cells combine to form organs to accomplish specific functions that serve the organism as a whole. Cells come in two kinds, prokaryotic and eukaryotic. Prokaryotic are simpler cells that do not have a true nucleus. Instead, prokaryotic cells house their DNA with all its genetic information in a poorly demarcated part of the cell. Prokaryotes occur as bacteria and archaea. Eukaryotic cells are larger and more complex and host their genetic information in a nucleus separated from the cytoplasm by a well-defined internal membrane. The cytoplasm in a eukaryotic cell contains a variety of well-defined parts called organelles, each having a particular function in the operation of the cell. Humans and all other animals, as well as plants and fungi, are eukaryotes. Reproduction in prokaryotes is asexual and usually takes place by binary fission. The DNA of a prokaryote exists as a single circular chromosome. In prokaryotes, the chromosome is replicated and the two resulting copies separate from one another due to the growth of the cell. The enlarged prokaryote splits in two halves, creating two identical cells or clones. This binary fission does not provide an opportunity for genetic recombination or genetic diversity, but prokaryotes can share genes by other mechanisms, for example, when infected by a virus, when in contact with genes found in the environment that originated from another prokaryote, or where DNA is transferred between prokaryotes by means of a pilus that attracts and forms a bridge between two cells. Therefore, there are mechanisms for more efficient genetic recombination within bacteria and archaea. Due to their overall simplicity, eukaryotes are thought to represent the first cells that developed during Earth's history. The metabolism of current prokaryotes is much varied, but as Earth's secondary atmosphere was almost entirely devoid of oxygen, early prokaryotes must have been anaerobic. During the anaerobic phase of life's history, living organisms could acquire energy only by fermentation. The energy yield from carbon dioxide fermentation is low, and the byproducts include alcohol. Alcohol is a high energy compound, and for the cell, a waste product that must be eliminated. The inability to use energy contained in alcohol puts limitation on the anaerobic cell. A large surface to volume ratio is required to allow rapid diffusion of food in and waste out. Anaerobic cells must therefore be small. Anaerobic cells cannot afford to deploy energy resources on the maintenance of specialized organelles, including a nucleus. Prokaryotes need free space around them, as crowding interferes with the movement of nutrients and water into and out of the cell. They also require space for the elimination of toxic waste products, alcohol, and in the early photosynthetic organisms, oxygen. Therefore, they live in a single cell or are strung end-to-end in chains. Prokaryotes cannot form the three-dimensional structures that are required for multicellular life. For at least 2 billion years, the only life on Earth was prokaryotic. Eukaryotes are more complex, larger cells, equipped with a clear nucleus and organelles, which are specialized structures within the cell. Mitochondria are a type of organelles where the biochemical processes of respiration and energy production occur. Eukaryotes strive in aerobic environments and use respiration for metabolism. Respiration is much more efficient than fermentation. As a result, eukaryotes can be much larger. Eukaryotes can specialize and form large multicellular organisms and reproduce sexually, which offers an efficient mechanism for genetic recombination, leading to faster evolution. Eukaryotic cells are the cellular unit of plants, animals, fungi, and protists. Forms of life are organized into groups according to their genetic and evolutionary relationships, called a phylogenetic tree. Species are organized into taxonomic ranks of 3 domains: bacteria, archaea, and eukarya, that comprise 6 kingdoms. Eukarya comprises the 4 kingdoms of animals, plants, fungi, and protists. The hierarchy is based on 8 major taxonomic ranks: domain, kingdom, phylum, class, order, family, genus, and finally, individual species. For example, our species of Homo sapiens is part of the genus Homo, the family Hominidae, the order of primates, the class of Mammalia, and the phylum of Chordata, and the kingdom of Animalia. We start the history of life on Earth in the Hadean, with the formation of the oceanic and continental crusts. The Hadean is formally defined as the period of Earth's history for which we do not have a rock record. Hence, by definition, we have no record of life in the Hadean. The early Hadean Eon is characterized by hellish conditions, with surface temperatures so high that water cannot be found in solid and liquid states. As the surface progressively cools, water floods into global ocean basins that will offer the conditions for the emergence of life. Meanwhile, intense volcanism feeds the atmosphere with carbon dioxide. The combination of water and carbon dioxide in the presence of energy spontaneously forms biomolecules that can be combined to form sugars, proteins, lipids, amino acids, the building blocks of life. Any early development of life must be interrupted by a giant collision between Earth and a Mars-sized planet called Theia. The collision vaporized the surface of Earth, and the debris gathered to form our moon. The collision took place about 20 to 100 million years after the solar system coalesced. Dating of the oldest rocks from the moon at 4.44 billion years ago indicates that this event occurred very early in Earth's history. Early life was very small and fragile, and it left little mark in the environment, less in the rock record. The only unmistakable microbial fossils are found in sedimentary rocks that formed in a shallow ocean environment about 3.5 billion years ago in the Archean. But life could have happened well before the end of the Hadean Eon, 4 billion years ago. So when did life arise? If life emerges early and often on any viable planet or moon, then perhaps there was a stable biosphere by 4.4 billion years ago, within Earth's first 150 million years. All the ingredients were there: oceans and air, minerals and energy. Giant impacts with asteroids and comets would have challenged the survival of this Hadean life, but life could have found harbor in deep sea hydrothermal vents environments, other volcanic zones, or even deep within marine sediments. Perhaps life came into being more than once during the Hadean, perhaps even many times, due to the ineluctable laws of organic chemistry. If so, then the 3.5 billion years old fossils would represent an ecosystem almost 1 billion years old already. If, on the other hand, the emergence of life is a difficult and long process, then a date closer to 3.5 billion years ago may be more likely, and the Hadean Eon was lifeless. In that case, those precious oldest fossil remains of the Archean Eon mark the true beginnings of the biosphere. Early life in the Archean and earlier is poorly documented in the fossil record. The earliest evidence of life are chemical signatures, microscopic filaments, and microbial mats. Carbon found in 4.1 billion-year-old zircon minerals have a chemical signature suggesting an organic origin. Other evidence of early life are 3.8 to 4.3 billion-year-old microscopic filaments from a hydrothermal vent deposit in Quebec, Canada. Unlike fossils, these chemical signatures and microscopic filaments are merely compatible with the presence of life, but they do not provide undeniable proof. The development of cellular life must have been the culmination of several incremental evolutionary steps. The first cellular live organisms must have been prokaryotic due to the relative simplicity of this cell type. Early prokaryotic metabolism must have relied on chemosynthesis, the synthesis of organic compounds using energy derived from reactions with inorganic chemicals, typically in the absence of sunlight. The production of complex molecules within living organisms or cells, called biosynthesis, must have become increasingly complex. Cells must have slowly developed the machinery needed for metabolism, and they have eventually found the means of reproduction and storage of genetic material through DNA and RNA. None of these evolutionary steps are independently documented in the rock record. However, there is evidence that the secondary atmosphere in the Hadean and Archean eons was poor in oxygen. So early life was prokaryotic and anaerobic. Anaerobic prokaryotes used fermentation for metabolism, generating CO2, water, and alcohol as byproducts. Prokaryotic life dominated the world for 2 billion years, from the Archean to the Proterozoic. The oldest fossil evidence for life dates back to 3.5 billion years ago in the Archean Eon. These first well-preserved fossils are microbial mats called stromatolites. Stromatolites are sedimentary rocks, a few centimeters to about a meter across, that formed where colonies of cells precipitated thin layers of minerals in shallow water settings. Microbial mats covered wide swaths of shorelines, consolidating sand in intertidal zones. Because the sedimentary rocks of stromatolites correspond to a shallow water environment, it is likely that these biodeposits correspond to unicellular phototrophic organisms, meaning that they obtain their energy from the sun. The paleo environment where stromatolites formed is well understood. because modern equivalent can be found, for example, in Shark Bay, Western Australia. Modern stromatolites are layered biochemical structures formed in shallow water by the trapping, binding, and cementation of grains in biofilms, especially by cyanobacteria. Modern cyanobacteria are a free-living photosynthetic prokaryotes that release free oxygen in the atmosphere through photosynthesis. Cyanobacteria are a more advanced form of prokaryotes with internal membranes where photosynthesis is performed. Since there is evidence for the presence of Archean rocks that could not form in an oxygenated atmosphere, it is likely that Archean cyanobacteria did not produce free oxygen. The presence of prokaryotic life therefore did not immediately affect the atmosphere. But Earth and its primitive microbial population were poised to make the most dramatic transformation in the history of the planet. Over the next 1.5 billion years, photosynthesizing microbes will develop a dramatic chemical innovation to release the highly reactive, dangerously corrosive gas called oxygen. Photosynthetic cyanobacteria, a type of prokaryote, dominated the world from at least 3.5 billion years ago to 1.5 billion years ago, from the Archean to the mid-Proterozoic eons. During that time, the photosynthetic process in cyanobacteria evolved to exhale oxygen as a byproduct of cell metabolism. As a result, much oxygen was released in the atmosphere, but it quickly rusted minerals at Earth's surface and became trapped in the geosphere. Exposed rocks were oxidized, and Earth's land surface looked like the surface of Mars today. It took about 2 billion years for Earth's oxygen sinks to be filled. And during this entire time, almost half of Earth history, the prokaryotes had the world to themselves. Eventually, an oxygenated atmosphere started to form, the so-called Great Oxidation Event. This major transformation created Earth's tertiary atmosphere, after the first atmosphere that was vaporized by solar winds following planetary accretion, and after the secondary atmosphere that was released by volcanism and protected by the magnetosphere during the Hadean. This drastic environmental change decimated the anaerobic bacteria, which could not survive in the presence of free oxygen. With an oxygen-rich atmosphere also came the ozone layer, which filters out harmful ultraviolet radiation, eventually allowing organisms to survive and flourish in shallow waters and on the land. Stratospheric ozone formed around 600 million years ago, just a few million years shy of the Cambrian radiation, when myriads of complex life forms evolved in a geological instant. We therefore owe our current atmosphere to the development of photosynthesis by cyanobacteria and their descendants. The Proterozoic eon is a 2 billion year period that ranges from 2.5 billion to 541 million years ago. During this time, most of the central parts of the continents had formed and plate tectonic processes had started. Photosynthesis by microbial organisms, such as single-celled cyanobacteria, had been slowly adding oxygen to the oceans. As cyanobacteria evolved into multicellular organisms, They completely transformed the oceans and later the atmosphere by adding massive amounts of free oxygen gas and initiating the Great Oxidation Event. Before the Great Oxidation Event, iron was floating in aqueous solution, but the release of oxygen forced iron to precipitate into minerals such as hematite and magnetite, leaving banded iron formations, marine chemical sediments, mostly between 2.5 and 1.8 billion years old, that consist of alternating layers of reduced and oxidized iron-rich minerals. Life evolved tremendously during the Proterozoic. After the Great Oxidation Event, organisms seem to have wasted little time in turning the lethal waste product, oxygen, to an advantage. While some prokaryotes develop oxygen tolerance through the adoption of specialized enzymes, a major change happened through the appearance of eukaryotic cells. With eukaryotes came two major innovations: the invention of respiration as a more efficient metabolic process, and the development of sexual reproduction, which enhanced genetic recombination and accelerated subsequent evolution. An oxygenated world changed the chemistry of the planet in significant ways. For example, iron remained in solution in the non-oxygenated environment of the earlier Archean Eon. In chemistry, this is known as a reducing environment. Once the environment was oxygenated, iron combined with free oxygen to form solid precipitates of iron oxide, such as the minerals hematite and magnetite. These precipitates accumulated into large mineral deposits with red chert known as banded iron formations, which are dated at about 2 billion years. The formation of iron oxide minerals and red chert in the oceans lasted a long time and prevented oxygen levels from increasing significantly, since precipitation took the oxygen out of the water and deposited it into the rock strata. As oxygen continued to be produced and mineral precipitation leveled off, dissolved oxygen gas eventually saturated the oceans and started bubbling out into the atmosphere. The Great Oxidation Event also triggered Earth's first glaciation event around 2.1 billion years ago, the Huron glaciation. Free oxygen reacted with methane in the atmosphere to produce carbon dioxide. Carbon dioxide and methane are called greenhouse gases because they trap heat within the Earth's atmosphere, like the insulated glass of a greenhouse. Methane is a more effective insulator than carbon dioxide. So as the proportion of carbon dioxide in the atmosphere increased, the greenhouse effect decreased and the planet cooled. As the planet cooled, liquid water turned to ice near the poles. Highly reflective ice at the poles reflected more sunlight into space, thus cooling Earth even more. In a rapid positive feedback loop, ice eventually covered virtually the entire globe, a situation called snowball Earth. By contrast, the end of each snowball Earth episode produced some of the most rapid and extreme changes in Earth's history. With almost all surface rocks insulated by ice, carbon dioxide from volcanoes built up to extremely high levels in the atmosphere. At a tipping point of carbon dioxide concentration, a greenhouse effect caused rapid heating and melting of equatorial ice, which raised temperatures further as more solar radiation was absorbed. This climate cycle of icehouse and hothouse periods occurred several times during the Proterozoic. Icehouse is accompanied with mass extinctions. Hothouse is associated with increased size and complexity of organisms. A large evolutionary step occurred during the Proterozoic eon with the appearance of eukaryotes around 2.1 to 1.6 billion years ago. Eukaryotic cells are more complex, having nuclei and organelles. The nuclear DNA of eukaryotes is capable of more complex replication and regulation than that of prokaryotic cells. The organelles include mitochondria for producing energy by respiration and chloroplasts for photosynthesis. The eukaryote branch in the tree of life gave rise to fungi, plants, and animals. Eukaryotes presumably developed through a symbiotic relationship with photosynthetic cyanobacteria via a process called endosymbiosis. One cell swallowed another cell whole. Normally, the bigger cell will simply digest the smaller, but in this case, the bigger cell incorporated the smaller one in a symbiotic relationship, perhaps in order to enhance its metabolic process. Another important event in Earth's biological history occurred about 1.2 billion years ago when eukaryotes invented sexual reproduction. Sharing genetic material from two reproducing individuals, male and female, greatly increased genetic variability in their offspring. This genetic mixing accelerated evolutionary change, contributing more complexity among individual organisms and within ecosystems. More than 600 million years ago, in the late Proterozoic, single-celled organisms learned how to grow and move as a single entity. They learned to become animals. The earliest unambiguous fossil evidence for an ecosystem that was dominated by animals, as opposed to microbes and algae, comes from the so-called Ediacaran period, a time interval that began about 635 million years ago, which was shortly after a snowball Earth event. The first of these rather distinctive symmetrical fossils to be recognized came from the Ediacara region in southern Australia. These fossils represent an array of soft-bodied animals, some of which are easy to imagine as distant relatives of jellyfish and worms. Closely related fossils have been found in many places around the world, in rocks from a 65 million year span between about 610 and 545 million years ago. The Ediacaran fauna offers a first glimpse at the diversity of ecosystem that evolved near the end of the Proterozoic. These soft-bodied organisms were among the first multicellular life forms. Ediacaran fauna did not have hard parts like shells and were not well preserved in the rock records. However, they were widespread in the Earth's oceans. The transition of soft-bodied Ediacaran life to life forms with hard body parts occurred at the end of the Proterozoic and beginning of the Phanerozoic eons, a period characterized by an explosion of biological diversity called the Cambrian radiation.